Welcome to Infection and Immunity Evidence Explained, a Doherty Institute podcast. I'm Rebecca Elliott. The 29th of April marks the International Day of Immunology, a day celebrated worldwide to raise awareness of immunology as a basis for individual health and well-being. Thanks to COVID-19, we understand our immune systems a little more, but immunology as a discipline is very complex. To commemorate this day and take a deep dive into the world of immunology, we are joined by none other than Laureate Professor Peter Doherty, the patron and namesake of the Doherty Institute. Peter became the first vet to win a Nobel Prize in Medicine in 1996, together with Swiss colleague Rolf Zinkernagel, for discovering how the immune system recognises virus-infected cells. Peter, welcome back to Infection and Immunity Evidence Explained.
Oh, great to talk, of course. Always great.
Now, the last time you were on this podcast was in the midst of COVID-19 when you had just published your book, An Insider's Plague Year. Now, does that feel like a lifetime ago?
It feels like a very long time ago, Rebecca. At that stage, I think there were certain certainties that seemed likely to come through, and they're less certain.
What do you mean by that?
Well, I thought we'd be able to get a vaccine, and we did, and we got it relatively quickly, and some people were saying we'd never get any sort of vaccine. And it seems we did get vaccines that, from all the evidence, tend to prevent the development of severe disease. But they don't stop people getting infected. And though they make the incidence of complications like long COVID lower, we think, they don't actually prevent it and give total protection. Very hard now to sort out what's going on with immunity to COVID because so many people have had it and so many people have been vaccinated multiple times. Well, so it's hard to know what the protective thing is because people can also have it without being aware they've had it.
Plenty of discoveries still to be made on COVID-19. And I guess that's a nice segue to what we're talking about today, which is a topic that you know extremely well, and that's immunology. So let's start with a definition. What is immunology?
Well, it's the science of immunity, the immune system that protects us from infectious agents. I think that's why it's evolved, to protect us from infection, but it also does other things too. It can often keep cancers in check, and particularly skin cancers. And of course, it can also have a downside because we can develop autoimmune diseases where the body reacts against itself. So it's a finely balanced system. It's one of the two great systems that we have for interacting with our external environment. The other, of course, is the central nervous system with the eyes and the ears and the brain and all the rest of it. The immune system operates below the level of consciousness, except when we get sick and we feel some of the consequences of that immune response. And because the immune response can make us feel sick. It's not hurting us, but it can make us feel sick. But it doesn't have a central processing unit like the brain, and it's a very dispersed system with cells all around the body. So it's a very complex system.
Yeah, it absolutely is. And you won your Nobel Prize for discovering how the immune system recognises virus-infected cells. So can you briefly provide us with an overview of what that process is?
We made a discovery way back in the 1970s, and we, as my Swiss colleague Rolf Zinkernagel, we were both very young then. I was 33, I think, when we made the discovery. I'd been doing science for about 10 years. He's a medical graduate. He came into it not so long before because he's younger, 5 years younger, and of course his medical training took longer. And we discovered that for a category of white blood cell that goes round in the bloodstream and monitors our body, a category of white blood cell called the killer T cell only recognizes virus-infected cells if they come from individuals of the same transplantation type. And that, what defines us as individuals, of course, is our winning smile and our wonderful personality and how charming we are. But biologically, what defines us is the spectrum of transplantation molecules we have on the surface of our cells. We have the potential to have 6 different transplant molecules on the surface of our cells. And that means that really means, unless we have an identical twin, if you're thinking of matching all 6, then basically we're all different. And as a result, when we transplant an organ like a heart or a kidney into somebody else, generally the person who gets that organ has to be immunosuppressed. They have to have a treatment that stops them rejecting the organ. So anyone who's had a heart transplant or a lung transplant will likely be for life on immunosuppressive drugs, or a kidney transplant, okay?
Yeah.
So everyone was very interested in these transplantation molecules, and there'd been an enormous amount of work done on mice to define them genetically and to isolate them in different mouse strains. But nobody actually knew what they were for. Why did we have a transplant system? What was it all about? Why had God in his wisdom, or her wisdom, designed a system where a mother could potentially, for instance, reject the fetus, or the other way around? And it turns out they're not really about rejecting transplants. What they are is a surveillance system, a system whereby our immune system can check out our living cells to make sure that they're all okay and they're not doing something really nasty, like becoming cancerous or being infected with a virus, which is what happens. Viruses only grow within living cells, or being infected with a virus and acting as a factory that can produce new virus particles. What we discovered is this works through those transplantation molecules. The killer T cells that go around in our body and are the hitmen of the immune system because they kill off damaged cells, recognize those cells as damaged because the infection or the cancer process changes those transplant molecules so they no longer look like part of us, they look foreign. The way that happens is a little bit of the virus binds into the tip of these molecules when they're being made in the cell, and when they're put on the surface of the cell, that molecule then looks different because it's carrying a bit of virus or a bit of cancer protein. And as a peptide. And that's how our immunological surveillance system works, which keeps our body in good shape. So this was an enormous discovery because nobody had even suspected that's what this transplant system was doing. That was what it was for. And it revolutionized that part of immunology, which is what's called the cell-mediated immune system. And it revolutionized thinking. We didn't understand it fully at the beginning, but we developed a theory around it, and the theory turned out to be right.
So how has your discovery then contributed to our understanding and development of vaccines and medicines and so forth?
Well, when you make a discovery like that, we made the initial discovery and it showed us something that was dramatically different. We didn't fully understand it because the science, the technology, wasn't really there at the time to do the right experiments. So over the next 10 years or so, thousands of people worked on this problem. You know, this is 2 young guys in Australia, working in Australia in Canberra, make this discovery. It's published and widely discussed. And we suddenly went from being totally obscure unknowns to being well known in the science community, which means you're not well known in the general community. You're still totally obscure as far as the general community, not as though you're someone important like a football player. And so basically, a lot of people worked on this, trying to understand what was going on. And it was worked out about 10 years later by several separate groups, in fact, using new molecular technology, what we call structural biology or X-ray crystallography. And it was finally defined that a little bit of the virus was being presented by these molecules. We'd called it altered self. They defined what this altered self was. So, that's where you get Nobel Prizes, for instance. You make the initial discovery, lots of other people work on them, and then you get the credit because they always go right back to the people who make the discovery.
So let's take a step back a bit and talk about what happens when we do have a pathogen presented to us. So what actually goes on? What's the process?
Well, it depends on the pathogen, but say it's a virus.
Yeah.
I mean, bacteria can grow outside our cells, and you've got intracellular bacteria and extracellular. Mycobacteria, for instance, the ones that cause tuberculosis, tend to grow within our cells. So the virus, but if it's a virus like the SARS-CoV-2 virus that causes COVID-19, or influenza virus, or hepatitis virus, or whatever, the virus has to first get into our cells, and then it uncoats and it takes over the genetic machinery of our cells to make new virus particles. So it becomes a virus-producing cell factory, and it can kill the cell as it goes. So if it's a cell in your lung that's infected, if it's one of the cells that's involved in gas exchange from the blood, which is, what keeps us alive, that we take in oxygen and we push out carbon dioxide, then that virus can potentially kill that cell. And influenza can kill you simply by infecting cells in your lung. And so what's important is to get rid of those cells as quickly as possible. And this is what the killer T cells do. It takes about 7 or 8 days, 10 days or so for that response to develop. But when it does, they'll knock off those virus-infected cells before too many get infected. Of course, if too many are already infected and these T cells come along and kill them off, they may kill you as well. So that's part of the problem. Everything is balanced in the immune system. So that once that it became understood that this was happening, they started to think about how to formulate vaccines so it can involve this type of response. But it's not the main part of the vaccine response. The vaccine response mainly depends on what we call the antibody side of things, and these are molecules that float around in blood that are made by cells called plasma cells, which develop from another cell type called a B cell. We'd understood that side of the immune system for much longer in a sort of broad general sense, but we didn't understand the T cell system until we made our discovery.
And so what happens when, we're exposed to a flu virus for the first time, for example, and then we're exposed again? Tell us about the memory of the immune system.
We get what we call seasonal influenza epidemics. What happens is the virus, like COVID-19, the SARS-CoV-2, flu viruses change, and usually about every year or so, new variants emerge, which will infect a lot more people again. So it's the same virus, but it's changed a bit, and it can escape that antibody immune response, and it can infect us again. But the cell-mediated, the T cell response, can be against other parts of the virus. The protective antibody response is against proteins that are on the surface of the virus, and it blocks that infection by binding to those proteins. They change by mutation, and that is an immune escape variant, as we call it. And that immune escape variant can then infect us again. But the cell-mediated immune response can be against any protein from the virus, the ones that are involved in replicating the virus, or the ones that are structural to the virus, and so forth. So it tends to be much more cross-reactive, but it doesn't give us immediate protection. So we think that with a virus like influenza, and to some extent with COVID-2, being infected stimulates T cell memory in the long term, and it's highly cross-reactive because these responses often don't change as much as the antibody one. That gives us some cross protection, but it tends to protect us against getting severe disease rather than against getting infected in the first place. It's the antibody response that protects us against getting infected.
Can you go a little bit more into the antibody response?
Well, the antibodies are they're molecules that are made by a cell called a plasma cell. These are big protein-secreting cells. They sit around, and the antibodies themselves are proteins that just float around in the blood. So if you think of the T cell responses like the hitman, he comes up and he doesn't have a gun, he just has a dagger. The hitman comes up, contacts the damaged cell, and then kills it, but it has to contact it. It can't do it from a distance. The antibody molecules are made by these plasma cells. They're secreted into the blood. Plasma cells are often sitting off in our bone marrow, and they grab hold of the virus. So they're, if you like, in a war terms, the killer T cells, like someone killing someone with a bayonet. The antibody responses are like a gas attack. The stuff goes everywhere. It grabs hold of the virus particle and then stops it getting into the cell. And these are the main vaccine protective responses, the antibody responses. And most vaccines are designed to try and make them preferentially, as is the COVID vaccine and the flu vaccine.
So, Peter, can you just tell me what's the difference between the innate and the adaptive immune system?
What we've been talking about so far, Rebecca, is the adaptive immune system. This is the highly specific immune system that targets something on the surface of a virus or a virus-infected cell. And it takes 7 or 8 days to develop. And the cells, everything is cellular, the cells that promote these responses are selected. They're very infrequent in the whole pool of potential immune response cells that we have. But because of that 7 or 8 day gap before we get that adaptive immunity actually working, what has to happen is the cells have to multiply and multiply and multiply, the right ones, and then they have to differentiate and start producing these antibody molecules or turn into these killer T cells. But before that works, we need something to keep us alive, and that's the innate immune system. And the innate immune system is a much less specific, broad type of response that activates all sorts of mechanisms early on, produces various molecules, some of which they're all made by cells, of course, but they get into our blood, they circulate in our blood, and they can, for instance, they're called cytokines or lymphokines or chemokines. They can, for instance, make us feel sick. Because if they're in the blood, they're flowing through the brain, they can interact with the receptors in the brain, which will give us our perceptions of how we feel. And so the fact that we get a fever depends on this innate immune system and the molecules that are produced during innate immunity. And they interact and they cause us to increase our body temperature. So the innate immune system is making a very broad response. A lot of the molecules that it makes, they're producing some fairly toxic molecules, in fact, that are designed to kill, say, an incoming bacterium, but they also can do damage to us. And you can actually get a phenomenon called cytokine shock, where the innate immune response is so powerful that it can make us very sick indeed.
What can we do if that actually happens? How do we reverse that?
There's a lot of research on trying to ameliorate that. It hasn't been tremendously successful at the moment. Shock can be can kill us, basically. And so, but there's a lot of focus on that. You try and neutralize some of these molecules that think may be involved. You can make antibodies that would block them, for instance, monoclonal antibodies. Some of the new therapies are of that type and so forth. And of course, some of those innate molecules are the ones that, for instance, are the final end pathway in some autoimmune diseases. So it's basically it's as though the adaptive immune system is individually targeted, very specific, very powerful. The innate response, much broader. It's a broad spectrum response.
You touched on autoimmunity there. Can you describe or just explain what that actually means?
Well, the immune system it has to learn what is us, self. Any individual's immune system has to go through a process of learning what is part of us and what is not part of us. Self and non-self we talk about. So people might think of those in terms of psychological discussion, but actually self and non-self, self and foreign is central to the discussion of immunology. And the place where the body basically tends to learn what self and non-self is in the T cells being processed through the thymus. But it's also possible, and we've known this for a very long time, that if you take, say, mice or rats and you, say, grind up the brain and you inject it with something, inject that stuff back into other rats or mice with what we call an adjuvant, something that boosts that innate immune system and gets the whole thing going, you can develop an autoimmune type disease. So if things are presented in the wrong way, sometimes people start to react against themselves. And this is generally, autoimmunity is generally more of a problem in women than men.
So let's use the example of COVID-19. Why do some people get a mild illness while others end up in hospital?
Well, in a general sense, if you have a defective immune system or your immune system is declining, you're much more susceptible. As we age, it seems we're much less able to deal with things that are novel. When we're young and our immune system is very healthy, we can deal with a whole lot of stuff. I mean, a young child, for instance, in the days when smallpox was a terrible, terrible pandemic virus and killed very large numbers of people, people would take children, or doctors would take children, and they'd practice what's called variolation. That is, they'd take dried material from the skin lesions. So, smallpox causes a horrible skin pustules and things. They'd take dried material from that, and then they'd scratch it into the arms of small children, and they get a bit of a fever and usually survive okay. And then they were protected. So when a child is young, they can often set up an immunological memory, which will more or less go for life or go for a very long time. And that's the basis of childhood vaccination. We don't normally get those childhood vaccines again, though we probably should as we get older. We probably should get boosted against things like whooping cough again, because there's now fewer children are being vaccinated, and everybody, especially as they get older, are at risk. So basically, you make extremely good immune responses when you're young. And then as you age, your immune responsiveness, especially for new things, gradually falls off. So that was what we saw with COVID. From about age 50, people became increasingly more susceptible. And the most susceptible, of course, are the very elderly who have really terrible immune systems. They're totally awful. My immune system must be awful. I'm 83.
Yeah.
So it's not too bad. But there is variation in older people about how good their immune system is. And the ones who don't have a very good immune system can be very susceptible. And then, of course, the other people who are very susceptible are the people who are either have some sort of immunodeficiency disease, or they're being treated, say, for cancer, which can kill off a lot of the antibody-producing cells and antibody-forming cells. So that's one category. But on top of that, people get severe disease, and we often don't really understand why some do and some don't. And that's true of all infections. If you take Japanese encephalitis virus, for instance, and this class of viruses, which recently came down onto the Murray River in Australia, most people who get bitten by a mosquito carrying that virus don't get very sick. They may get a bit of fever, but they're likely okay. But a proportion, the virus gets into the brain and they develop what's called encephalitis, and it's a very severe disease. So we don't really understand why that variation occurs in many infections.
So we're often told that certain foods or vitamins can boost our immunity, that the marketing companies tell us that. Is there any truth to this? We'll have the answer to that question in a moment, but just a quick break to say that if you're enjoying this podcast, why not share it with your family and friends, or rate and review it on your favourite podcasting app? And now back to the show.
It's hard to know if there's any truth to it, because what is obvious is a good, balanced diet is important to protect you, and that children and people in developing countries, for instance, under conditions of malnutrition, are much more susceptible to infections of various sorts. But apart from that, the stuff that this will be, vitamin C was supposed to do wonders for us. And there was a big thing about vitamin D during COVID. You said it's about marketing. A lot of it is about marketing. As someone said, you can take, if you've got a good diet, if it's a good balanced diet, you're getting protein, you're getting some animal protein as well as plant protein and so forth. And you may not absolutely need animal protein, but you do need some vitamins and stuff. But if you've got a good balanced diet, as someone said, taking multivitamins, for instance, is a way of making very expensive urine.
So you're saying potentially stay away from the vitamins, but balanced diet.
I would say be very skeptical about the claims that are made.
Yeah.
I mean, you see all these things about boosting your immune system.
Yeah.
I'd like to see some evidence.
Yeah, right. Okay. There you go. You
But it
heard it
doesn't
here.
mean they don't, but it's just the evidence is not there on
The
the whole.
evidence is not there. So we've heard the term herd immunity, thrown around a lot over the past few years. What exactly does herd immunity mean, and what role does it play in preventing disease outbreaks?
Well, for instance, herd immunity is a good concept with something like measles, okay? It's more complicated with COVID because COVID can infect people who are substantially immune because the virus changes so quickly. It's not that it's changing much more quickly than flu, but I think what it is that it binds. It's able to change in ways that escape the immune response more easily than flu does. So, but herd immunity with measles, the measles vaccine gives you protection. Kids are protected against measles if they get the vaccine. They won't get measles. Or if they do, later on, they'd get something very, very mild, and they probably even would be aware of it. And in fact, early on with measles vaccine, it was realized that the vaccine needed 2 doses. If they just gave one, they needed to also give a booster to bring the level up. Otherwise, you get breakthrough infections. But if most people are vaccinated against measles, most children are vaccinated against measles, then babies won't get it. You can't vaccinate babies with measles vaccine, okay?
Why is that?
Because it's a live virus vaccine, you've got to wait till their immune system develops a little. So basically, if you don't vaccinate all the children, then babies are at risk. And that's herd immunity, basically, that the majority are vaccinated, they're not getting infected, they're not spreading the infection. Therefore, those people who are vulnerable, babies, people who are immunosuppressed and so forth, will also be protected simply because the virus is not circulating at any sort of level. The same is true of whooping cough. Now that I had my earliest memory, I think, is of having whooping cough. I remember struggling for breath, and I remember that whooping cough sound, whoop, whoop, sort of sound. I can remember that, and I think that's the first thing I remember. I had it. I've never been an athlete, and I think that's possibly because I had lung damage at that stage. It's a terrible disease, and it can be fatal. But fewer and fewer people now vaccinating their children against whooping cough. As a result, there's more whooping cough around. So even if you're elderly or older and you've had those vaccines as a child, it's probably a good idea to get a booster later in life because you can get whooping cough again.
Now, we've got the International Day of Immunology coming up on Monday, the 29th of April, which is a few days away. Why is a day like the International Day of Immunology important?
So these things are about trying to get messages out to the broader community. And it's, as we all know, it's not that easy to engage the broader community. The best way to do it would really be to have Bruce Springsteen do something on immunology. Or Taylor Swift.
Well, yeah, Taylor Swift would definitely, if she could sing a song about immunology, that would be a win.
Well, she'd certainly appeal more to the demographic that has small children.
Yeah, for sure. So we've touched on a little bit of ageing and immunology. And that's the theme for this year's Day of Immunology is immunity through the ages, navigating the science of ageing and immunology. We've talked about what happens to our immune system as we age. Is there any way of reversing any damage to our immune system as we age? Can we reactivate it?
There's a lot of interest in that, but I can't point to anything that actually is really working. It's the process of aging is there are aspects to it that may be reversible, and a lot of people are interested in that sort of area. But it would be the same principles applying to other cells in the body. I would think. Basically, all sorts of systems in our body wear or run down as we get older. It's just a fact of life. I'm acutely conscious of it at age 83.
What excites you still about the field of immunology?
I think modern molecular technology has all sorts of possibilities. Many of those are tested out on almost an ad hoc basis, and particularly in humans. I think where immunology is going now is much more. I mean, immunology, when I started out, the really intellectual front of immunology was experiments in laboratory mice and so forth, where you could do nice defined experiments and you could get good results. Very hard to do a lot in humans because, firstly, of course, with a mouse, you can get cells from the spleen and the lymph nodes, the places where the immune response, the spleen, lymph nodes, particularly where the immune response happens. If you're getting lymph nodes out of a human, it's just because maybe you've got a biopsy or something where a bit of lymph node comes out. So mainly human immunology, you're looking at what's in the blood. If you're a bit more sophisticated, maybe you might look at what's in the fluids you get from the throat or from the lung, but mostly it's what's in the blood. So, but what excites me now is the enormous potential for really understanding the human immune system much better, because with new modern molecular technologies, we can take a white blood cell out of the blood. If it's a killer T cell, we can tell, and we know what it's specific for. We can tell what activation state it's in, what molecules are being read out, what it's making, what it's doing. And I think that will give us much, much greater insight into the aging process means for the immune system. At the moment, we're going through a stage where using those types of approaches, looking at what's happening in human cells, and we can do that with very, very small amounts of blood, which means we can often do things in babies, for instance, that we couldn't have done previously because you just can't take very much blood from a baby or a small child. But now, small sample sizes, we can actually do a lot. So I think we're going to understand the human immune system over the next 20 years. We'll work through and we'll understand what aging means, what the neonatal immune system is like, much, much better than we do now. And that will hopefully lead to new discoveries and new insights. So that's where a lot of immunology is going. That doesn't mean we don't need to do experiments in mice, because often when you make a discovery and you think you've identified the role of a particular molecule, then you can go back and say, manipulate mice genetically and so forth and do experiments that are very useful and give insight. But obviously, the study of the immune system that we're interested in is the study of the human immune system. Or if we're a farmer, maybe the veterinary immune system. But it's not so important because, most of the animals that we're interested in, unless they're pet animals, tend not to survive very long. They don't get old.
So if there was one great mystery of the immune system that you would like to solve, what would it be?
Of the immune system, how it counts. I don't know how it counts. I don't know how it... We maintain relatively constant numbers of cells in the blood. We greatly expand those numbers of cells for a specific immune response during an infection, and then it comes back to a moderate level. I'd like to know how it actually counts. And we don't even know how big the human immune system is at any time. We know what's in the blood. If we've got to take a mouse, we know what's in the lymph nodes. But there are all sorts of lymph nodes and all sorts of white blood cells, for instance, sitting around in body tissues and all sorts of places. Because the immune system, unlike the nervous system, doesn't have a central processing unit. It doesn't have a brain. It's often hard to know just where it is numerically. I mean, post-mortem, people have been taking brains out forever and weighing them. You can't do that with the immune system.
No, you can't.
No. So how does it count? That's what I want to know.
All right. Well, take that on notice to our young immunologists. And what advice do you have for our young immunologists out there?
Well, science has changed from the time that I started doing it. We made a very big discovery that somebody would have made eventually. I mean, it would have happened. And because discoveries tend to happen when technology gets better and you've got a better approach. And we were using the latest techniques at that time, which now seem very primitive, but it allowed us to make this breakthrough. But basically, now it's all much more, we're really almost drowning in information at the moment. It's sometimes very hard to pick your way through it and to go to the particular molecule or the particular type of event that then needs to be studied in depth. And so there's a tremendous potential, but the problem is that where there was often one tree standing and you could identify it easily, now it's a forest. So I think For young scientists, my advice is always to look very closely at the data you're generating, think about it, just don't go on and do more and more experiments. Try to work out what it's telling you. I mean, what we're doing in science and in immunology, in all science really, but in immunology, is we're interrogating nature and trying to get answers back. And if you think about it that way, you're instructed by nature. We are instructed by nature.
Thank you so much for joining us back on the podcast, Peter. It was great to talk to you today.
Thanks, Rebecca.
And thanks to all of you for listening to Infection and Immunity Evidence Explained, a Doherty Institute podcast. As part of the Day of Immunology, we'll be holding a discovery tour at the Doherty Institute on Monday, the 29th of April at 4:15 p.m. Registration is free but essential by visiting immunology.org.au/events. That's immunology.org.au/events. If there is a specific topic you would like us to cover, please get in touch with us by emailing doherty-media@unimel.edu.au. That's doherty-media@unimel.edu.au. If you've enjoyed our podcast, please rate and review on your favourite podcasting app and subscribe if you haven't already. The Doherty Institute is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. See you next time.